Welcome back to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from neuroscientists across the schools and departments of the University of Cambridge, reflecting the pioneering work and diverse interests of our members here. Each seminar focuses on one of the new interdisciplinary themes of Cambridge Neuroscience, and for more info on the talks covered in this seminar series and all things neuro-related here at Cambridge, please follow us on Twitter at CamNeuro and follow the links below. So good afternoon, everybody. Welcome to the Cambridge Neuroscience Interdisciplinary Seminar Series. And we're back for our new term, and we're delighted that Professor Amy Milton has joined us today. Amy is the Professor of Behavioural Neuroscience at the Department of Psychology here at Cambridge, and she's also the Ferris Willits Fellow in Neuroscience at Downing College here in Cambridge as well. So today she's going to talk to us about targeting maladaptive emotional memories to treat mental health disorders, giving insights from rodent models. So we're delighted to have you, Amy, and we're really looking forward to your talk.
Brilliant. Well, thank you very much, Dervila, for the invitation to come and speak today, and thank you all for joining, despite the Adrian seminar going on at the same time. So I'm going to be talking about some of the work that my lab has been doing over the last couple of decades, actually, looking at maladaptive emotional memories. And we are particularly interested in these from the perspective of developing new forms of treatment for mental health disorders. So we're really interested in the contribution that these memories make to the persistence of mental health disorders like post-traumatic stress disorder and drug addiction. And more recently, we're becoming also interested in disorders like obsessive-compulsive disorder. So really, we're kind of at our core, I suppose. My lab is a memory lab, and we're really interested in a process known as memory reconsolidation, which I'll be talking about in a few slides' time. And reconsolidation exists, and we think it's evolved to be an adaptive kind of process by which memories can be updated, but it gives the potential for targeting old and well-established memories, including these maladaptive emotional memories, to develop new forms of treatment for these mental health disorders. Now, of course, that's highly experimental, and although there are some studies where these reconsolidation-based interventions are starting to make it into small-scale clinical trials, the majority of the work so far, and the majority of the work that I'll talk about today, has been proof of principle work in rodents. So I'll also be giving some insight into what rodent models can contribute to this. So first of all, kind of the motivation for this work is really that there is a strong clinical need, an unmet clinical need for more and better treatment for mental health disorders. So it's estimated that Europe-wide, 38% of the population will experience a mental health disorder during their lifetime, and that of that 38%, 35 to 50% of those with mental health problems won't be receiving any form of treatment. Even once people have entered treatment, relapse is common, even when treatment has initially been effective. And actually, even where treatment does work, we don't necessarily have very clear indications of why our current therapies work, which then limits our ability to optimize those and to make them effective for the people that aren't currently being well treated by them. So although it's not a dire situation, there clearly are treatments that work, there is a clear need for treatment innovation, particularly for those who aren't responsive to therapies that are currently available. And one such innovation, or one process that's been proposed as being a new treatment innovation, is this process of memory reconsolidation. So reconsolidation was a theory of memory first actually proposed in the 1960s, but it really got going again around the year 2000 with the work of Karim Nader in the lab of Joe Ledoux at NYU. And reconsolidation theories are often best understood by contrasting them with more traditional views of memory. So I'm sure that many of you are familiar with the idea that we start off with short-term memories that can become consolidated into long-term memories. And what the reconsolidation view does is it doesn't necessarily disagree with that. Memory consolidation occurs after an experience has been had. But rather than thinking about memories as existing in short-term stores or long-term stores, the reconsolidation view instead argues that we should think about memories in terms of their activity state. So memories can exist in an active state, which is short-lived and unstable, or they can live in this kind of long-term, persistent, inactive state. And so following their initial consolidation, memories enter this inactive state where they are stable and persistently stored. But a key difference between the traditional view and the reconsolidation view is that under the right conditions, according to the reconsolidation view, memories can be induced to re-enter the active state, even long after they've been initially acquired. And so through this process of reactivation, or more mechanistically, destabilization, the memory returns from this really stable state to a state where it now becomes once again malleable and labile. Now, normally that would occur under conditions, and I'll talk about the boundary conditions in a few slides' time. This would normally occur under conditions where the memory would be updated. So new information could be incorporated into the memory while it's in the active state. And then the memory is thought to reconsolidate or restabilize back into the inactive state in its updated form. But one of the prominent sort of kicking off of the new reconsolidation period, the study by Karim Nader, demonstrated that actually you can block this process. And so if we take an old, well-established memory, and he took a Pavlovian fear memory, we induce it to go into the active state, but then instead of allowing this memory to reconsolidate, we block that process. Now, they happen to use a protein synthesis inhibitor called anisomycin, which they administered directly to the basolateral amygdala. It blocks synaptic plasticity there, but you can equally use other agents as well. NMDA receptor antagonists or even beta-adrenergic receptor antagonists also seem to be effective. But if you induce this memory to enter the active state and then you block this reconsolidation process, then actually it should be possible to trap that memory within the active state where it's transient and unstable. And depending on how strongly you take this reconsolidation view, it should then be possible to then persistently weaken, or in the strongest versions of this theory, erase that memory so that it should no longer have any impact on behavior. And so this single or very few interventions, this combination of behavior plus pharmacological intervention leads to a persistent reduction in the impact of that memory on behavior to the point where it doesn't look like it's having any impact at all. Now, it didn't take long after this rediscovery of reconsolidation in 2000 for people to start considering that this could be a useful approach to disorders that have a component of maladaptive emotional memory to them. So Barry Everett and I argued for this from the perspective of drug addiction. Also other labs like Jane Taylor's lab have made similar points. Merrill Kint has argued for the use of these kinds of treatments for PTSD, as in fact have Adrian Ledoux themselves. So there is real interest, but we do just need to take a moment to pause to think about some of the potential challenges there are with these reconsolidation-based therapies. It's now been 20 years and I have quite a lot of people ask me what's taking so long for these therapies to get to the clinic. And actually, there are some things that we do need to consider. The majority of the work to date has been done for Pavlovian memories, but we know that instrumental memories are also important in these mental health disorders. And much of the work has been done with adaptive models rather than maladaptive models. So what I'll be doing over the course of the next half hour or so is talking about some of the attempts that we've made, and some of this is preliminary data. So, I will caveat some of the conclusions that I'll make. But what we've been trying to do in terms of moving to more translationally relevant models in the rodents, thinking about how that might interact with boundary conditions, and then thinking about how these boundary conditions might differ between Pavlovian memories and instrumental memories. So the very kind of learning theory lens of looking at these rodent models, where we're trying to sort of deconstruct these more complex mental health disorders, then try to reconstruct those mental health disorders and see the effectiveness of these reconsolidation-based therapies. So I'll begin with Pavlovian memories, sort of as the simplest place to start. And in terms of Pavlovian memories, I think that PTSD really has been to some extent considered sort of the prototypical maladaptive memory disorder, particularly because it can be, as it's been argued since the mid-80s, be conceptualized as a form of aberrantly strong and generalized Pavlovian learning. So if we think about Pavlovian learning, I'm sure that most people in this virtual room are familiar with this, but I'll just recap for those who are less familiar. The key aspects of Pavlovian learning is that if you take a stimulus that is initially motivationally neutral, so like an auditory stimulus, for example, and present it to an animal, of course they won't react. But if it's paired with a motivationally relevant unconditioned stimulus, so in this example, the pairing of an auditory stimulus and a mildly aversive foot shock, this will then lead to a subsequent conditioned response to that cue on its own, because it's acquired both predictive and affective emotional value from that pairing with the unconditioned stimulus of foot shock. And it's been argued that you can make this analogy for PTSD as well. So if we take a combat stress example, the cues associated with the traumatic event, so the sound of gunfire, for example, when paired with the threat of serious injury or death, leads to, on the battlefield, an adaptive response of fear. But because this memory is aberrantly strong, because it's overly generalized, this then leads to reactions to similar stimuli, these trigger stimuli, like the sound of a car backfiring, in situations where that's no longer appropriate. So, for example, when the veteran has returned home. So we can we can accept, I guess, at some level that this may be the case, but there is, I think, there are some key differences between the kinds of adaptive fear learning that you see for Pavlovian fear memories and more maladaptive fear learning, which is what we're seeing in PTSD. And so some of our more recent work has been trying to move to these more translationally relevant models that try to capture some of those maladaptive aspects of fear learning. And one model that we've been particularly interested in is one that was originally developed by Mike Fanslow's lab, the stress-enhanced fear learning model. And essentially the way that this works is that animals are put through a massed shock exposure in a distinct time and space compared to the rest of the behavioural procedures, but they're put into a specific context where they're presented with massed foot shock, unsignalled foot shock, which is meant to be an analogue of the trauma. And then you can subsequently see that having been through this traumatic experience, some of the animals will be susceptible to this and some will be resilient. And that's actually true for the patient population as well. Many people will go through a traumatic experience, but not everyone will subsequently go on to develop PTSD. Now, the way that we assess the impacts of being resilient or susceptible in this model is to look at the impact that that phenotype has on subsequent fear learning and extinction of fear learning. So what you can do with this population, now we happened in this study where we were trying to refine the procedure, we left 24 hours between the mass shock exposure and looking at the effects on subsequent learning. The Fanslow group have done this 3 months later and you still see the same effect. So it seems to be a very nice translational model. But essentially what we did was to take that population of animals, give them contextual fear conditioning, standard contextual fear conditioning, put them through fear extinction, and then look at the recovery of the memory subsequently. And what you see in this procedure, and we did the traditional Fanslow 15 shock procedure and then a more refined version where we gave fewer shocks, you see that the animals that have been through the shock procedure, as compared to a no shock control who are just exposed to the context rather than the trauma itself, all of the animals show increased fear conditioning. But the animals that are susceptible show delayed extinction, so impairments and extinction compared to the resilient animals, and that's consistent with what you see in the human population as well. And they also show greater recovery of fear when you test them subsequently. Now, this becomes really important from a clinical perspective because it's not necessarily the case that the conditions under which a memory will become unstable is the same when that memory is adaptive versus when it's maladaptive. So we know that memories don't destabilize every time they're retrieved. And in fact, there are boundary conditions that determine whether a memory will destabilize or not. Now, we know quite a lot about these boundary conditions. A lot of this was worked out in crabs. So we know that, for example, prediction error and surprise, so the violation of expectations between what's expected and what actually occurs, seems to be one of the really important boundary conditions within getting memories to destabilize. This was first demonstrated in surprised crabs, but has subsequently been replicated with surprised rats and surprised humans as well. So it seems to stand across species. And from the perspective of reconsolidation being about memory updating, of course, that makes complete sense. If there's prediction error, that suggests there's something that needs updating with that memory, and therefore that memory should return to the active state where it can be updated. But we also know that the relationship between prediction error and memory destabilization isn't monotonic, and actually it's quite complicated. So in work that we did a few years ago with Emiliano Merlo, we showed that there's actually two separate memory processes that can be engaged during a re-exposure session, depending on the extent of the prediction error or the amount of prediction error that the animal is re-exposed to. So if we give the animal a small amount of prediction error, so in this case, Emiliano was using auditory fear conditioning, so one presentation of one tone at reactivation induces a little bit of prediction error because it's not shocked at reactivation. And but actually, if you give an animal repeated re-exposure, so in this case, Emiliano happened to use 10, this doesn't engage reconsolidation at all. It actually engages an entirely separate memory process known as extinction. And this is the formation of a new inhibitory memory that would then suppress subsequent fear. Now, Emiliano was particularly interested in what happens in between. So if you give 4 CSs or 7 CSs rather than 1 or 10, how would that affect the memory? Like, what's the transition and the relationship between these two separate memory processes? And this is a complicated set of data. I'm not going to go into detail. It's all published, so you can have a look at the original paper. But the key take homes from this are that we can see differences in the engagement of these memory processes depending on the extent of re-exposure. So at 1 CS, that seems to engage reconsolidation, and we can test this by blocking the dominant memory process by giving a drug like MK801, which prevents reconsolidation or extinction from occurring. So with 1 CS, a brief re-exposure means that MK801 reduces the subsequent fear memory, which is consistent with a blockade of reconsolidation. But if you give MK801 following extensive re-exposure, then what you actually do is block the formation of the new extinction memory, and you see enhanced fear when the animal is exposed to that long re-exposure session. Now, importantly, in the in-between conditions, for 7 CSs, that's just basically extinction. The animals behave pretty much as they do for 10 CSs. But in the middle, at 4 CSs, what we have is this, what Emiliano termed the limbo period, where the memory doesn't reconsolidate and it doesn't extinguish. And we know that's not a population level effect. It's not that some animals are doing one and some are doing the other. Actually, if you look in the brains of these animals as well, you can see that the reconsolidation window has closed, but the extinction window has not yet opened. So actually, the relationship between prediction error and the memory processes that are engaged is actually quite complicated. So we start off with retrieval. So when there's no prediction error, we're just retrieving the memory. A little bit of prediction error will give us reconsolidation. We then enter this limbo period where any manipulation would be ineffective because we're not engaging either process. And then we go into this extinction phase where actually, if we gave a drug that blocked synaptic plasticity, we'd actually make things worse. And what we don't know is whether the boundary conditions that have been worked out for these adaptive fear memories are actually the same for maladaptive fear memories. It's not a given that a maladaptive fear memory will behave like an adaptive one. So we have very preliminary data, and I will caveat this by saying this is very preliminary data. But essentially what we've done is take our animals that have undergone stress-enhanced fear learning, and we have done a contextual version of Emiliano's experiment. So we've given them, rather than sort of the standard fear extinction manipulation that I described first of all, we've given them re-exposure for 3 minutes, 10 minutes, or 30 minutes, which in adaptive fear memory studies, so in just standard contextual fear conditioning, would normally engage reconsolidation. 10 minutes would normally engage the limbo period, and 30 minutes would normally engage extinction. And to test which process we are engaging with each one of these re-exposures, we're also giving half of the animals MK801 and then NMDA receptor antagonists, which would block whichever process is currently dominant. And when we take rats from our 13 shock conditions, so they've been through their mass shock exposure, we separate them out into resilient and susceptible animals. We actually do have some preliminary data suggesting that there may be differences in the way in which the memory is behaving with these different re-exposure conditions. So if we think about the resilient animals, first of all, actually what we see is that we don't see an engagement of reconsolidation with a 3-minute re-exposure, which is what we'd see with the adaptive memory in animals that haven't undergone this stress-enhanced fear learning procedure. And in fact, these animals appear to be extinguishing by 10 minutes because MK801 leads to greater fear subsequently when they're tested. So these are all data from the retention test. And so that would suggest that they're actually engaging extinction far earlier than you would expect in the animals who don't have any trauma exposure. And 30 minutes looks like roughly the same pattern. But by contrast, if we look at the susceptible animals, again, 3 minutes doesn't seem to be doing very much, but actually this isn't statistically significant because we don't have the power. But if anything, it looks like the 10-minute re-exposure might instead be engaging reconsolidation, and we may be hitting the limbo period at 30 minutes. So that may suggest that we actually have a shifting of the boundary conditions with the maladaptive fear memory as compared to the adaptive fear memory. And that will have really clear implications when it comes to developing these treatments for the clinic, that we can't necessarily rely on lab studies, even human lab studies of adaptive fear memory. We need to think about how we can adjust our reactivation procedures to engage these more maladaptive and persistent memories. So even for Pavlovian memories, where this is meant to be the easy part of the equation, there's clearly work to be done. And things get a little bit more complicated still when we start to think about the interactions between these Pavlovian memories and instrumental memories. And we've done most of our work looking at this in the context of drug addiction. So like PTSD, Pavlovian memories do contribute to addiction as well, and they're particularly important for relapse. So people, places, and paraphernalia that are associated with a drug high subsequently come to have very powerful effects on motivating ongoing drug-seeking behaviour, but also precipitating relapse subsequently. And there's a whole host of studies now showing that actually, if you scan the brains of individuals who are currently abstinent from drug use, and you look at the reactivity of the brain, the responsivity, the amount of activity you get when you present them with drug-associated cues in the scanner, you can actually predict who will subsequently go on to relapse a few months later, because these cues have such a powerful impact on drug-seeking behaviour. But although these Pavlovian cues do clearly influence behaviour, they're not acting alone. I mean, a really important part of addiction, of course, is drug-seeking and drug-taking behaviour. So there are instrumental memories, whether we're thinking about someone ordering alcohol at a bar or a rat working in a self-administration procedure, pressing a lever in order to gain access to drug. The acts of drug-seeking and drug-taking are instrumental in nature, but of course they occur in the presence of these Pavlovian cues, whether that's a context or whether that's discrete conditioned stimuli. Bottles of alcohol, for example, are very good conditioned stimuli for alcohol use. Or, of course, in our operant chambers, we use more sort of circumscribed stimuli like lights and so on. But essentially, these instrumental and Pavlovian memories are interacting with each other, and in quite complex ways again. So we know that there are at least 3 psychologically and neurobiologically distinct ways that these cues can interact with each other. So these cues can influence relapse behaviour just simply by supporting approach to themselves. These cues become attractive, they become they become capable of supporting conditioned approach in their own right. And often that approach to the cue will bring the individual into the vicinity of where they can make the instrumental response to drug seek. We also know that these cues can bias memory retrieval towards drug seeking or reinstate the relevant motivational state. Whether that's the subjective state of craving or not is sort of debatable, but we know that they can assert this effect through Pavlovian conditioned motivation. And we also know that these drug-associated cues can maintain responding over delays and support new and flexible drug-seeking behaviour by acting as conditioned reinforcers. So we actually have 3 psychologically separable, and in fact, to some extent, neurobiologically separable routes to relapse, if we're thinking about this in an addiction context. And so one of the things that we were particularly interested in determining is whether it would be possible to disrupt these routes to relapse separately and also together in order to have a longer term effect on drug-seeking behaviour. So I'll start with separately and then move on to putting it all back together. So we began actually by looking specifically at conditioned reinforcement. And this was a procedure in which animals were trained to self-administer cocaine over an extended period of time. So animals were receiving hundreds of pairings in which they would they would nose poke in order to receive a cocaine infusion, but also presentation of a light. So they had a Pavlovian cue as well that became associated with the drug. And then the memory was reactivated in a very brief memory reactivation session in which they were presented with that light cue again, under the influence of either a vehicle solution or another NMDA receptor antagonist, AP5, directly administered into the basolateral amygdala. And then we subsequently then, after that single treatment session, took the animals and tested them repeatedly for whether they would learn to work for the cue in its own right. So if the cue can act as a conditioned reinforcer, animals will learn new and flexible seeking behavior for that cue in its own right. A little bit like the human example of money. Money has no primary reinforcing value, but we will learn to perform tasks in order to gain access to money. So if the cue means anything to the animal, they should learn to work for it. And what we found was that we only treated the animals once, but I'm showing the 2 groups separately. Here's the groups that had previously received the AP5, so that's the NMDA receptor antagonist, all groups that had previously received the vehicle. And these are the same animals tested repeatedly up to 29 days after that single treatment session. And what you can see is that while the animals that were that received the vehicle solution, if anything, increased the amount that they will work for that drug cue over time, the animals that received AP5 just don't ever seem to increase their responding at all. And in fact, we were also able to show by having a parallel group of animals in which we took the brains at the end of the shortly after the memory reactivation session, we were able to show that actually this was exerting its effects through the action of an immediate early gene, which is known to be critical for reconsolidation to occur. So we saw reductions in the expression of a gene called DIV268 only in the brains of animals that had undergone memory reactivation and received the AP5. So consistent with this idea that we're blocking the reconsolidation of that memory and having a long-term impact on behavior. So we also looked actually in a more kind of translational, using a more kind of translational approach. So administering an NMDA receptor antagonist directly to the brain is clearly going to be a challenge for clinical translation. But actually, there were good reasons to suspect that other drugs that could be administered systemically might also have similar effects. So we also looked at a beta-adrenergic receptor antagonist called propranolol. And this is already used in the treatment of hypertension. It had already been shown to block fear memory reconsolidation. So it wasn't a giant leap to see whether it would also block the reconsolidation of these cue drug memories. And what we found across 2 different classes of drugs, so for both cocaine and alcohol, essentially the same setup as I just showed you in the previous experiment, but this time animals were either working for cocaine or for orally administered alcohol and receive an injection, systemic injection of propranolol in conjunction with reactivation, and we see broadly the same effect. So the animals that receive propranolol don't increase their responding in the same way that vehicle-treated animals do. And interestingly, in work done by my graduate student, Moritz Schramm, we can show that this is centrally mediated because animals working for propranolol, animals working for alcohol-associated cues but treated with propranolol show the same sort of reduction in behavior. But if you administer nadolol, which doesn't cross the blood-brain barrier but has all the same peripheral effects as propranolol, you don't see that long-term reduction. So it must be centrally mediated, even though we're giving a systemic treatment. So for conditioned the ability of a drug-associated cue to act as a conditioned reinforcer, both NMDA receptor antagonists and beta-adrenergic receptor antagonists can disrupt the reconsolidation of those memories. But I told you about 3 routes to relapse. So what happens if we look at those other ones? And so we've done that. And to summarize, about 5 years work in 2 slides. Essentially, what we found was that if we administered NMDA receptor antagonists in procedures in which we were looking specifically at memory's underlying conditioned approach, or procedures where we were looking at conditioned motivation, in both cases, the NMDA receptor antagonist impaired the memory. So for conditioned approach, their behaviour returned to how it was on the first day of training. They didn't discriminate between a cue that had been paired with the drug and a cue that hadn't. And in the case of conditioned motivation, they actually didn't respond any more in the absence of the cue than they did when the cue was present. So what we have here, this dotted line represents equal levels of responding in the presence of the cue or the absence of the cue. Anything above that line is an enhancement in responding when the cue is presented. And what you can see is that with the error bars here, this doesn't differ at all. Now, by contrast, when we ran propranolol on the same experiments, we actually didn't find any effects on conditioned approach or conditioned motivation. So there does seem to be a dissociation in these neurochemical systems in terms of the different processes that they're able to impair. And that becomes important because if we start to think more translationally, so we start to think about all these different ways that these drug cues can influence relapse behavior. Actually, if we take a more translational model where an animal is self-administering a drug using the same instrumental response that he'll use after training to then drug seek subsequently, actually all 3 of these routes to relapse can act simultaneously. So if an animal is trained to self-administer cocaine, for example, by pressing a lever, and then he also gains access to a light when he receives his cocaine infusion, that behavior can be supported by Pavlovian conditioned approach because he's approaching the lever and he's approaching the location of the light, and that brings him into the vicinity of where he can make that drug-seeking response. But once that light is presented, that can then support the reinstatement of the motivational state to work for cocaine through Pavlovian instrumental transfer or conditioned motivation. And actually in the test sessions, we test animals in extinction, so to just assess their drug-seeking behavior free of any contamination of the drug. And actually their responding is primarily supported by the fact that the cue acts as a conditioned reinforcer. So actually in these more translational procedures, all of these things are acting simultaneously. And of course, in reality, for real patients, this is also going to be the case. And so actually it turns out that then if we do a version of our reconsolidation blockade experiments using this kind of procedure, we allow them to self-administer, give them a brief memory reactivation session, and then subsequently test them for drug-seeking behavior on the same instrumental response, what we see is a dissociation between what the NMDA receptor antagonist can do and what propranolol does, such that in the presence, when they've been treated previously with an NMDA receptor antagonist, their responding is reduced to a level that actually doesn't statistically differ from not presenting the cue in the test at all. So this is a group that receives saline during the memory reactivation session, but then don't have the cue at test. And there's no statistical difference, although they're slightly numerically higher, between this group that receives MK and do see the cue at test and this group which don't have the cue at test. And these are significantly lower than vehicle-treated animals. But by comparison, animals treated with propranolol don't differ from those vehicle-treated animals. And we think that's because MK801 can disrupt all 3 of these routes to relapse simultaneously, and we've shown that separately, whereas propranolol is only acting on conditioned reinforcement, leaving 2 separate routes that could support the behavior subsequently. So for both PTSD relevant memories and addiction relevant memories, we can disrupt the reconsolidation of Pavlovian memories that contribute to the persistence of those disorders. But what about the instrumental memories? Because I've just very proudly shown you on that graph that this group are lower than this group. But actually, when we think about the instrumental memory itself, so what we have here is the amount of lever presses they're making on the lever that used to give drug versus the lever that's never given drug. And although it's true that the Pavlovian cue isn't potentiating that lever pressing, it's clear that all of the animals still discriminate between a lever that used to give drug and a lever that never gave drug. So the instrumental memory actually hasn't been targeted in this procedure. But that might be because instrumental memories need different conditions in order to destabilize. And actually this comes onto a related question, which I think hasn't received as much attention in the reconsolidation literature as it might have done. And that's where the expectations in the violation of expectations comes from. So if we think about prediction error being really important to make a memory destabilize, that implies there was an expectation to be violated. And so actually we really need to think about how those prior expectations are generated. And so you can imagine a situation, for example, where a prior expectation is there, but a new experience is perceived as being sufficiently different that it's actually irrelevant to the previous memory. And so you wouldn't actually update that memory. Or you might have a situation where you have a prior expectation, but actually the experience that the animal has in the reactivation session is just so random that actually it's not reliable enough that you would update a previously existing memory. So it's really only under very specific conditions, perhaps where the prior expectation is a little bit more imprecise, or there's an interaction with imprecise sensory input, that actually this might drive this kind of updating process. And so work done by Geoff Lee and his lab at the University of Birmingham seems to support these kinds of ideas. And we've done a bit of a replication and a bit of extension of that, which I'll talk about now. So these are all for adaptive. Again, this is all preliminary data, so this is ongoing work. But these adaptive instrumental memories do seem to have slightly different boundary conditions to the Pavlovian memories. So our typical Pavlovian memory reactivation would involve re-exposing animals to the cue in the absence of any reinforcement. Now, if you do that for instrumental memories, actually you don't see any effect of your manipulation. So just not reinforcing reactivation for the animals appears to be too different as an experience, and they just don't update. Now, Geoff Lee's lab had already shown actually that if you take an animal that's been trained on a very predictable reinforcement schedule, so no Pavlovian cues, just responding to sugar pellets on a fixed ratio 1 schedule, very, very predictable. And then you reactivate them on a different fixed ratio schedule, so you put them onto an FR20, that doesn't seem to lead to any updating of the memory. And we replicated that when we looked at this fairly recently. And what we think is happening there is that the animal has an expectation, they've got an experience that is clearly very precise, but is clearly different to what's gone before, so the original memory remains intact. But interestingly, if you put the animals onto a slightly more unpredictable schedule, so if you put them onto a schedule in which they're receiving on average the same amount of reinforcement as in this schedule, but it's more varied, that actually seems to be a bit of a sweet spot where the more imprecise input at reactivation makes that memory more unstable. But you can't go too mad on making it imprecise, because if you put them onto a random ratio schedule where the information is now really imprecise, on average they're being reinforced as often as they are in these two conditions, but it's completely random, then you lose the effect again. So there actually seems to be this really delicate interaction between those prior conditions and the reactivation, the prior expectations and the current information that's coming in determining whether the memory will become unstable or not. And we've also managed to show, so work, there's actually a molecular correlate to this in the brain. So work done by Harry Robson when he was an MPhil student in the lab showed that if you look at a molecular marker of memory destabilization, so we know, for example, that specific postsynaptic density proteins, so one that we looked at was SHANK, shown here back in 2008 actually, to decrease as the memory destabilizes and then to then return to baseline levels once that memory has restabilized. When Harry was looking at that in the brains of the animals that had undergone these procedures and comparing that to animals in the predictable version versus the unpredictable version, he actually found a selective reduction in SHANK expression in the brains of animals that had undergone that unpredictable reactivation in the anterior dorsolateral striatum. So there seems to be a molecular correlate to these memories destabilizing. But as I said, preliminary data, we haven't yet causally assessed whether activity within the anterior dorsolateral striatum is necessary for the reconsolidation of these memories. So that's all adaptive work. And I'm now sort of just going to finish up by thinking about where we would really like to go with this as we go into maladaptive instrumental memories. And we've become particularly interested more recently in obsessive-compulsive disorder as an example of compulsive-like behaviour that we think actually overlaps to some extent with the compulsive-like behaviours that are seen in disorders like addiction and compulsive eating. And we've been looking at this in a really highly translational way in a collaboration with Sharon Moraine-Zamir's lab at Anglia Ruskin University, using a task that was developed by Dawn Eagle here in Cambridge called the Observing Response task. Now, this is a fully translational task. It was designed to be possible to use in both rats and humans, but it is a new task, so I will just take a moment to explain how it works. Essentially, what we're doing is asking the individual to respond, in the case of the rats, on one of two levers, in the case of humans, to stimuli on the screen. And one of these responses will be reinforced with rewards. So for the rats, it's sugar pellets, for humans, it's points. But the one that is reinforced is quite difficult to determine because the schedule of reinforcement is quite unpredictable, and we change which response is correct randomly through the session. So we can make this more or less difficult. Obviously, we start with an easier version of the task and then we make it more difficult for the animal. So essentially what you end up with is, two levers, any one of which could be correct at any point in time. But to help our individuals, to help our participants and our rats on this task, we give them the ability to check which lever is currently being reinforced. So on the back of the chamber for the rats, or on a separate button for our humans, there is a checking response where if the rats press the lever, they'll switch a light on above the lever that's currently being reinforced, or they will highlight the stimulus that's currently being reinforced in the case of the humans. Most individuals will use this information adaptively. So they will check, they will then respond on the response manipulandum that looks like it's correct. When the reinforcement rate seems to drop below a certain level, they'll go back and check again and then carry on working on the responses being reinforced. But a subpopulation of our animals show what we refer to as dysfunctional checking. So they continue to press this lever on the back of the chamber, even though it does nothing. It doesn't make the light stay on for any longer. It's never been paired with reinforcement. Actually, they would be far better off directing their behavior to the lever that's going to earn them sugar pellets, but they continue to check on this dysfunctional lever, on this checking lever. And so we're referring to that dysfunctional checking as something that we think is akin to the kind of dysfunctional checking behavior that's seen in the checking subtype of obsessive compulsive disorder. And so we have seen we have a hypothesis essentially that we're testing in this work that clearly, the route into OCD clearly does rely on aversive memories. There is an anxiety that is driving the initial start of the behavior. But actually, we think that that process of being the dysfunctional nature of the checking is probably more akin to something like the habitual responding that you would see in a disorder like drug addiction. So we think that this is kind of indicative of an over-reliance on habits. In fact, we have some evidence that this might be the case, because a sort of a way of classifying animals, we know there are certain subpopulations of animals that are more likely to rely on habits, and one such subpopulation are sign trackers. There's an extensive literature, which I won't review now, but there's an extensive literature showing that if you compare animals in their response to Pavlovian cues, in this case, the cue is a lever. If you just literally present a lever and then give them reinforcement, so it's a fully Pavlovian schedule, some animals will spend a lot of time really engaged with that cue, and some animals will go to the goal location to go and retrieve the reward. And we can classify animals as being sign trackers, who are more kind of cue bound, and goal trackers, who are less cue bound. And actually, we know from the addiction literature that animals that sign track seem to be more susceptible to the influences of these Pavlovian cues on drug-seeking behaviours. So they're more likely to continue seeking drug when it's no longer available. They more quickly transition into compulsive, habitual and compulsive drug seeking. Then they're also more sensitive to cue-induced relapse. In humans, you can assess something similar to this sign tracking by doing attentional distractor tasks and essentially assessing how much does a cue that was previously reinforced distract you from a task that you're trying to do now? And you can do this using measures of eye tracking. And interestingly, people who score very highly on sign tracking in these tasks are also more likely to endorse in questionnaire measures addictive behavior and obsessive compulsive relevant behavior as well, suggesting that there seems to be a link between sign tracking and these kind of more habitual behaviors. And we've actually seen in our previous work that these sign trackers are in fact the animals that show high levels of dysfunctional checking. So if we compare goal trackers and sign trackers compared to control animals as well, who don't undergo any auto-shaping, we can see that sign trackers show increased levels of dysfunctional checking, particularly when we make the task more uncertain. If we put them under conditions of perceived threat, so we put them in an anxiogenic environment, you see an increase in checking from goal trackers, but that's all functional checking. The dysfunctional checking is still primarily sign trackers. And interestingly, we put them in a situation where they're under actual threat. So if they get it wrong, they not only miss out on reward, they actually have the risk of getting a foot shock. All animals will increase their checking, functional checking under those conditions. But again, it's only sign trackers who show high levels of dysfunctional checking. And in fact, when other groups will return back to baseline when the shock is removed, sign trackers continue checking at really high levels, suggesting there's a rigidity to that behavior. So actually, we think that there may be something here, that essentially these aversive memories are kind of the route in, and that's probably more about functional checking, but that the over-reliance on habit, you can probably use sign tracking as a marker of that. And so the work that the lab is now doing is attempting to tie together these data with our sort of more reconsolidation-based approaches. So we're really interested to know whether individual differences in other measures of cues on instrumental behavior correlate with dysfunctional checking. That's work being done by Felipe Espinelli-Amarin in the lab with the rats, but also Shira Berg and Emily Brees at ARU, Shira's at Cambridge, in humans, and that's collaborative work with Sharon Morin-Zamir. We're also testing whether sign trackers are more likely to show compulsive dysfunctional checking, and the relationship between, the causal relationship between sign tracking and dysfunctional checking, but ultimately all to end up, hopefully, in a few years at the goal of working out if we could target the reconsolidation of the Pavlovian and all the instrumental memories that support this behavior, to then try to develop new forms of treatment for obsessive compulsive disorder as well. So very forward looking to finish up on, but I'll just conclude by hopefully saying that what you've seen is that there is clearly a new including PTSD and addiction and OCD, that a really kind of key aspect of this, at least in my opinion, is collaboration across disciplines, across species, as perhaps using psychological processes as a framework for facilitating this translation. And that understanding how these sort of adaptive and maladaptive Pavlovian and instrumental processes, although it's very basic reconsolidation mechanistic research, actually then potentially allows for new treatment development. So I'll just then finally finish by thanking all the people who've contributed to the work, but also all of you for your attention as well. And I'm really happy to take any questions.
Thank you, Amy, for a wonderful seminar. Join us next time when we welcome Professor Timothy O'Leary. Timothy is a professor of information engineering and neuroscience in the University of Cambridge, where together with his group, he studies the nature of information processing, learning and intelligent behaviour in living systems. He teaches engineering, edits for a major open access journal, and directs several international projects. His current research aims to understand how information is represented in the brain and how these representations evolve in time, and how we can leverage control engineering principles to understand variability, diversity and robustness in nervous systems. Tim will be speaking to us about feedback control in the nervous system, from cells and circuits to behaviour. For more info on the talks covered in this seminar series and all things neuro-related in Cambridge, please follow us on Twitter at CamNeuro and follow the links below. See you next time.